Okay, this is prep 2 for lecture 27. This is the third time that I have tried to record this. There's been many interruptions. And so we finished just talking a little bit about the layered structure of the brain. And I'm just pointing out here that the spinal cord, you'll remember the ventral versus dorsal specification that we talked about. And the same kinds of things are going on. So you still have progenitor cells, stem cells, and they're giving rise to different kinds of neurons and so on. So the same kind of thing is happening. On the dorsal side, a lot of the cell bodies that you see here are for commissural neurons, and they're shown here in blue. And so the cell bodies are up mostly in the dorsal sort of lateral region. But the interesting thing about these ones is that their axons, the extensions, they extend down and around across the midline, and they connect on the other side. And part of their role is to coordinate information on both sides of the body. Now, there's structure in the brain as well, where the two different hemispheres of your brain connect with each other. And it's very important, actually, for things like your eyes. The information you're taking in one eye and the other eye, they have to be coordinated properly in the brain. And the same is true for any sensory information that's coming in. And the coordination of motor control and that sort of thing. So those are the commissural neurons. On the ventral side, the nerves that are developing, most of the cell bodies are in what is called the ventral root ganglia. So it doesn't show it here, but when this is more mature, there will be a process, or more than one. There'll be a big bulge, and that's where all the cell bodies are. That contains the nucleus and most of the cytoplasm. And then there will be processes, sometimes more than one, and that's where the axons are moving out. And these are the motor neurons on this side. So they are making their way out to innervate the muscles of the body, so that you can move your arms and your legs and twist your back around if you're looking at something, etc. All this kind of thing. And then there are sensory neurons on the dorsal side. And these are actually derived from neural crest cells. So the neural crest cells have migrated down, and then they've come through the somites and connected back to the neural tube. And they are forming part of the peripheral nervous system, but these are sensory nerves. And so they are going to be connecting to sense organs in the body, and they're conducting information towards the spinal cord. And the information is being conducted away from the spinal cord in the case of the motor neurons. So just keep in mind, ganglia is plural, ganglion is singular. So if you have one, there's a ganglion, and then if you have a whole bunch of them, they are ganglia. And I'll show an image of the sort of more mature nervous system in the actual lecture, so you can see what that looks like. I'm just checking to make sure there's nothing I was going to mention that isn't included. Okay, so we're going to talk now for the rest of the time about the neural crest, how it's kind of specified, and then what it ends up contributing to. So neural crest is a temporary population of cells, and these are stem cells. They are multipotent, which means they can form many types of cells, and they arise from the borders of the neural plate. So just a reminder that the neural plate, we covered this in a previous lecture, but the neural plate develops when you inhibit especially BMP signaling, but to some extent Wnt is in there. I haven't really talked about it. BMP is the one I focused on. And then if you have FGF signaling. And then the presumptive ectoderm has Wnt and BMP signaling and delta too, but we really focused on BMP in the class. And so, but what you have in the region between the two, oh, and remember that there are neural cell adhesion molecules being expressed here, and then there are epidermal cell adhesion molecules being expressed here. That accounts for the fact that the two epidermal regions find and connect to each other to make one continuous epidermal, and the ends of the neural tube come around and bind to each other with those homotypic, remember, interactions. The regions, the two areas of boundary between those two types of tissues is not quite like either of those tissues. So the cells next door, the neural cells, have had BMP repressed, and FGF is very active. And then the ectoderm, epidermal cells nearby have plenty of BMP signaling. And so what happens is these guys get sort of a small amount of BMP signaling compared to the neural plate. So they're already predisposed to have different cell fates than either of these two tissues. And then as the neural tube is forming and the edges of the tube are trying to sort of find and bind to each other, these neural crest cells, they start to delaminate. They lose the connections between them. Remember, these epithelial layers tend to be like, layers of cells that have these strong connections between each other. In order for them to delaminate, to move out, they have to lose those connections and move as single cells. So you'll see a population of cells right in this region, but then they start to separate out and move, and they tend to migrate down dorso, from the dorsal side, down the lateral side towards the ventral. So they stay put in terms of anterior versus posterior, but they move down and around, and they are going to see lots of different organs and tissues, and differentiating into lots of different cell types. There's a number of different models for what causes the delamination, exactly whether it's signaling or changes in cytoskeleton or cell adhesion and so on, and probably some aspects of all of these are correct. So if you read further on this, if you happen to be interested in it, you will probably read multiple theories, and just be aware that a lot of the researchers that, at least that I've been sort of skimming the papers, that suggest that there's probably multiple processes going on that lead to the delamination, the separation away from the other two tissues, and then their migration. Okay, so in the early stages, the neural crest region is here, and then individual cells move down, and they move through somites to form ganglia. So this is sort of a schematic of the dorsal root ganglion, and also to connect to the autonomic nervous system, the sympathetic nervous system. This is the part you don't control consciously. And then individual cells also move and innervate other parts of the body, including the adrenal gland. So they are forming part of the adrenal gland. That is a mesodermal structure. And they're innervating the gut. Some of them are forming pigment-producing cells that are contributing to the skin. Others are going to be forming part of the heart, so myocytes, some of the muscle cells, those are mesodermal fates. And things like bone and cartilage of the face. The cartilage and bone is a mesodermal derivative as well. So notice that even though these are derived from ectodermal cells, we see different cell fates. We see some mesodermal fates as well as a lot of neural fates here. So the neural crest cells move. They don't stay where they are. They have to move to new locations to participate in producing heart and adrenal glands and innervating the gut and so on. The pigment cells have this dorsolateral movement, which is sort of across the back and down the side. So here's our embryo, and this is our dorsal side. It's a full embryo, and they move from dorsal towards the ventral down the side, which is what dorsolateral refers to. And they tend to stay just below the skin, like you're not seeing them wandering around sort of more internally. And if there's a somite here, they basically move over the surface. They don't touch the somites or enter them or go through them or anything like that. And they go across the whole part, so the entire somite, which I'm only mentioning because the other kinds of migration that occur is more specific than that. So other kinds of migrating neural crest cells will actually move over into or through the anterior somite, but not the posterior. So if this is the somite, and say this is towards the head and this is towards the tail, and the neural crest, suppose this is on the left side, the neural crest cells, they're here and they're migrating down. If they migrate across or through the anterior part of the somite, they just keep going. If they encounter the posterior part of the somite, they are repelled and they move either towards the anterior of their own somite, or if they're kind of way down here and they're towards this region, they get repelled, and they're quite likely to end up migrating across the anterior of the somite behind them. So there's a real preference for kind of the front half of the somite. And there's some experimental evidence that shows that it's some property of the somite itself that leads to this. And so I've made just a very simplified sort of set of somites here, and the migration pattern, which is across through the anterior part of each one. But if you do an experiment where you very carefully excise one of the somites and then you rotate it 180 degrees so that the anterior and posterior are reversed relative to the other somites, and then you allow the embryo to heal. Embryos have an amazing ability to heal from this kind of damage, and embryonic tissue that's damaged, when it heals, there's never scars, which is really interesting. But then when you look at the migration of the neural crest cells, they go across what would have been the anterior of that somite. So anterior posterior information is already on the somite, so that when you reverse its orientation, the neural crest still migrates across what would have been that original anterior. So there's something about these somites that, something different in the anterior versus the posterior part that leads to this migration. And there's a number of different molecules involved. For example, you may have heard of, if you took cell biology courses, semaphorins, and they're part of this process. But I just want to talk about the ephrin system this time. And so posterior somites have ephrin B1, which is a ligand, and the neural crest cells are expressing FB3, which is a receptor. And so they have very similar seeming names. So the convention is to write out the whole of ephrin for the ligand and just EPH for the receptor as a way of indicating which is which. The anterior part of the somites are also expressing the receptor. So when a neural crest cell with the FB3 receptor is migrating across here, the other cells have receptor for the same ligand, but it never encounters the ligand, so it's able to just move across and through very easily. But when the cell is moving into or onto or through the posterior part, it's going to encounter this ligand very quickly and there will be a repulsion, and then it will move. It'll keep migrating until it finds an anterior, either the anterior of the same somite, and sometimes the closest one is actually the next somite over. And so that's how that works. And now some of you may have heard of efferent signaling before. Efferent signaling is interesting because it often is bidirectional. So what that means is you've got these two cells, they're both moving, one has the ligand, one has the receptor, they find and bind each other, and then they both have a response to that binding that leads to the two cells moving away from each other. So both cells have an internal response after they connect with each other. And that happens often with this type of signaling. But in the case of the neural crest cells, you have somites that are pretty solid blocks of tissue at this point. And the neural crest cells are the ones that are actually migratory, moving cells. And so in this case, it's what we call unidirectional. So the binding occurs, but then there's only a response in the cell with the receptor, and that's the neural crest cell. The cells of the somites don't move at all. They stay put. Okay, and so neural crest cells, as I've said before, they migrate dorsolaterally along the AP axis, which means that they stay in their same anterior-posterior position. So if you've got cells that are originating here, they're migrating down and around here, and they'll contribute to these branchial arches, some of which form the jaw and parts of the face and so on. The cardiac region, they'll migrate down and around, and they'll contribute to different regions and so on. So that's what we mean when we say along the AP axis. We mean that the neural crest cells here are going to wind up innervating different cells along their route to the ones sort of in this position or further up. And this is just a list. I think I said in the learning outcomes, it would be good for you to just learn three of these. And there's some interesting ones. So the cartilage and bone of your face and skull, odontoblasts, so they contribute to formation of teeth, melanocytes, pigment cells, but myocytes, heart cells, and just different kinds of the cells of the adrenal medulla. Here is a typo that I just noticed on this figure. And then parasympathetic ganglia of the gut, parasympathetic, sympathetic, so parts of the sympathetic and parasympathetic nervous system. One thing to note is that some developmental biologists consider that the neural crest cells constitute a separate fourth germ layer. They're only found in vertebrates, but they do take on cell fates that are both mesodermal in some cases and ectodermal, and they're super interesting as well because there are some genes that affect neural crest development, migration, etc., that are thought to be associated with domestication of dogs, cows, and all different organisms. So I might assign a bonus assignment in that area. I just put this in here because I wanted to remind you, I'm pretty sure this will have been, well, I know this has been covered even in high school and stuff, but I just wanted a reminder because sometimes we don't think about these things, the year after or 2 years after. So the parasympathetic and sympathetic nervous systems are part of the autonomic system, which is the part that you don't sort of consciously control. And the sympathetic is sort of the emergency freak out kind of system where, you've got a rapid heart rate, your pupils will dilate, take in as much light as possible, see as well as you can, stop digesting because that's not important when you're in an emergency. And then your breathing accelerates and so on with the aim of getting more oxygen to your brain, making you more alert and aware, and letting you run away or fight back in some case of emergency. And the parasympathetic system is when the emergency is over, your heart rate decreases, your pupils, they constrict a little bit, you get to go back to digesting your food and stuff, and your breathing slows down. It's not that you can't affect these things, because a lot of these things you can control a little bit with things like meditation and breathing exercises and so on. But under normal circumstances, we're not ever, you don't have to remind yourself to breathe, right? It just